Friday the 14th is the last day. So now you're all going to get my grade determination form, so you can give yourself a barometer for how you are doing. So you all, I think most of you understand this, right? This is familiar to you. So one column is for the grades you earned, one column is for the grades you have yet to earn. You're welcome. There you go. You're welcome. So you can, again, for what's left, you can estimate, and when you estimate, it's usually the idea, unless you think it's something you're definitely at 100% on, like journal reflections. Oh, I'm sorry, the journal reflections come in default as zeros. I don't know why that is. Brightspace is just not a nice thing. But you didn't get a 0. I just didn't grade it yet. So don't worry about that. But like you might say, okay, I'm going to do all those points for the journal reflections. I'm going to do all 4 of those. You know, I feel pretty strong about the lab report. You know, I'll say like a 90, so you do 90% of those 80 points. Experiment design project we're going to start working on tomorrow in lab. We'll do reproductive anatomy for part of the lab, and then we'll spend the rest of the lab working on that. And you'll be posting in a discussion board, so you'll get points for several different aspects of that experiment design project, which will include an oral presentation with your group. So that's a couple of different activities. 80 points is the total amount for that. And then the final exam. So you add up what you earned, points that are left, and we add those together as total points for the semester. You can divide that by 10 to get the percentage. All right, let's proceed for that. I also did starfish reports last week, so again, I did that for everybody. Everybody got a starfish report, no matter what. So again, I'm not singling anyone out. That's just to help communicate with you, you know, anything that I saw as I was reviewing your grades so far. All right. So next up is exam number 3, which is next Monday. It's through where we finished today. So wherever we stop today, I'll let you just draw a line in your notes. And I have the last 2 materials. So hopefully we'll get through a pretty decent chunk of digestive anatomy. And I've already posted a bunch of videos on Brightspace, so I'll just remove some of those based on where we stop. And I'll edit the learning outcomes for exam 3 also to reflect where we stop today. Don't forget, assignment number 6, which is the nephron diagram, is due by the end of the day today. So that's one where you can just take a picture of it, turn it into a PDF, and then upload it to Brightspace. Or if you have it and you just want to give it to me before you leave class, you can do that too. But if you want to turn it in by the end of the day, you can just submit it right on Brightspace. And then assignment number 7 will be like our previous pre-exam assignment, so that'll be posted later today. It's on all the materials. It'll be on exam 3. It's open book. It's not timed, matching, fill in the blank, and all those kinds of questions. And it's due before class on Monday. Those are all my announcements, I think. I'm trying to remember. Anything else? Questions before we start? Okay, cool. So today we are going to start our next body system, which is the digestive system. So we'll be starting with digestive system function. We'll do just kind of a very general overview of what is going on in the digestive system, and then we'll get through the structures that allow those functions to occur. So we'll start out with the first part of the digestive tract, which is the oral cavity. Okay, to begin, we'll start out by looking at function. And the digestive tract is made up of basically a tube that starts at your mouth. Right, so this tube starts at your mouth and it ends at the anus. And this is a just continuous tubular structure. So from where your esophagus is to your stomach, you can see maybe the size of the tube changes a little bit. Maybe some parts are more like a pouch, other parts are more twisty. But it's all just one continuous linked structure from mouth to anus. So we'll look at some of the major functions that occur here. The first one is ingestion. So this is what's happening in the mouth, right? You ingest nutrients in the form of the food that you eat, and that is how you are first bringing nutrients into your body, through this process of ingestion. So this is just like eating. And then after you eat something, right, it doesn't just stay in your mouth. That would be not very helpful. The next thing that happens is that you move it out of your mouth and down into the next area of your digestive tract. So the second function is motility. So all throughout the digestive tract, we're constantly moving the material that you've eaten through different parts of the digestive tube until we move it all the way out of the body. So that's the second major function, is motility. All right, we also have, as a function of the digestive system, secretion of materials from the organs of the digestive system into the areas where the food is. So you can you think of anything that we would secrete, maybe from like our stomach or our intestines or our mouth onto food? Saliva. Yep. What else?
I'm so sorry.
That's okay. Think about your stomach. Like what kind of pH is in your stomach? Acid. So like stomach acid, right? Talked about amylase last 2 weeks ago in lab. What's amylase? What kind of a chemical? What is it? It's an enzyme, right? So we secrete enzymes that are involved in breaking things down. So secretions occur in lots of different places, right? We already named a bunch of them. So most segments of your digestive tract are secreting something. And what that secretion material is depends on what the organ in that part of the digestive tract is specialized for. So again, some examples were like saliva or stomach acid or enzymes, right? These are all things that could be potentially secreted. Okay, so then the next function, after we ingest something, we move it through the various tubular structures of the digestive tract, we secrete things on it. Our digestive tract is also involved in the process of digestion, right? So for our purposes, digestion is going to be considering breaking things down into smaller pieces. So we're digesting, we're breaking materials into their components. So there are two main types of digestion that happen in the digestive tract. These are mechanical digestion and chemical digestion. So mechanical digestion is where you are physically breaking food into smaller pieces. So think about like when you're chewing food with your teeth, you are mechanically digesting that food. You're physically breaking it into smaller physical pieces from the one solid piece that it was. And that kind of mechanical digestion, it starts in your mouth when you chew material up, and it continues throughout most of the muscular digestive organs as well. And then we also have chemical digestion, which is where we're taking the molecules that make up the food that you're eating, and we break those molecules into smaller molecular fragments. So mechanical is breaking things down into smaller pieces primarily to increase surface area. Chemical digestion is now we're taking the molecules of the things that you've eaten and breaking those into smaller fragments. So we can do our next step, which is absorption. So absorption now is the opposite of secretion, right? So for secretion, we're taking things from glands or cells in the linings of our digestive organs, and we're putting it on the food that's passing through. Now in absorption, we're taking materials out of the digestive tube and we're putting them into our bloodstream. So we're literally absorbing nutrients out of what was in our food and putting those nutrients into our bloodstream. Sometimes we put them into our lymphatic system first, but eventually everything winds up in the blood. So for things to get absorbed, as you may imagine, they have to be pretty small. You're not going to absorb an entire molecule of starch because starch molecules are enormous. So that's why things like chemical digestion are really important to this next process of absorption, because you can't absorb things unless they're pretty small. Okay, so after we've ingested something, we pass it through our digestive tract, and we've secreted things on it, and digested them, and absorbed a lot of nutrients, we take what's left, and we eliminate it. So no matter how hard we try to absorb as much as possible by digesting things down into small pieces, there's always going to be some material that we just aren't able to absorb or break down into a form that is absorbable, and that material is eliminated as feces. So that's the last process, is just getting rid of that unabsorbed material, and that way we make space for new stuff to come through that can, re-experience this whole process. All right, so any questions about any of these functions? All right, so what we'll do next is, for the digestive tract, we will primarily focus on this tube of the GI tract, the gastrointestinal tract. And we'll just kind of track our food from the beginning to the end. And along the way, if there are any accessory glands or organs, I'll point them out and we'll go over those as well. So we'll start out with the oral cavity, also known as the mouth. And the mouth is lined with an oral mucosa, right? So we have mucous membrane and all of the body structures that are open to an outside environment. In the mouth, we specifically have a non-keratinized. So remember, we have like the keratinocytes in our epidermis, then we have that really protective layer. So in the mouth, the types of squamous cells we have, they don't have keratin. So we call it a non-keratinized stratified squamous. This makes up the oral mucosa. Also in the oral cavity, we have, as you've seen in lab, when we were looking at some of these structures, we have a hard palate, and the hard palate is hard because it is made up of bone, and this separates the mouth from the nasal cavity. So, I know it sounds gross, but if you went straight up through the hard palate of your mouth, you would wind up, I'll put NC, you would wind up in the nasal cavity. And you can see here in the sagittal section that the hard palate is supported by the maxilla bone and the palatine bone. So that's what I mean, it's like a bony structure. And then the soft palate is soft in that it's not supported by bone. And the soft palate separates your oral cavity from your nasopharynx, right? The part of your throat that kind of goes up towards your nasal cavity. So I'll put NP here, right? Just to remind us, this is the nasopharynx. Okay, so it's hard palate, soft palate. And then just a review, right, hanging down off of the soft palate is the uvula, which we also talked about when we were doing the respiratory system, because the uvula helps to prevent food material from getting from your oral cavity and your oropharynx when you're kind of swallowing something. Keeps that food from going up into your nasopharynx and then into your nasal cavity where we don't want food to go. So that's part of the soft palate. And then, do I have it labeled on here? I feel like I don't. I'll just label it really fast. And then also, last major structure that we have is the tongue. Before we start talking about teeth, obviously we have those too. So there's our tongue. And the tongue does a bunch of stuff. The tongue, right, does a bunch of things. it manipulates food and then it moves the food around your mouth so you can chew it with like different parts of your teeth. It helps to push the food to the back of your mouth and towards your pharynx when you swallow. It also secretes a couple of different things, such as enzymes that help aid in digestion, even when you're just chewing the food in your mouth. Chemical digestion is already starting, as well as mechanical. All right, so I think those are all the major structures for the oral cavity that we can see very superficially. So next we'll get into the teeth, and then we'll start talking about salivary glands. But so far, any questions? Yeah.
What is this soft palate teeth made of?
That is a good question. I know it has some muscular components. I think that like parts of the soft palate that form those pharyngeal arches, like on the sides, do contain muscle that helps to kind of like contract and push food into the pharynx, like once your tongue gets it towards the back of your mouth. But other than, and it's definitely covered in epithelial tissue. I don't know if there's anything else besides muscle and epithelial tissue, but I'll check it out and see. Yeah. Other questions? Okay, so next, teeth. So we saw teeth already in lab, so we'll just review these. The teeth are adapted in our mouth to do different things with regard to the food that we eat. We'll kind of go through, we'll look at where these are, their names, and what their functions are in eating. So this is a mouth that's open, so we get, let's see, starting from here, it's like the hinge where the jaw would be. So this way is anterior, like towards the front of the mouth. And then this way is also anterior. So the incisors we find in the anterior part of the mouth. We have 4 incisors. And the incisors we use to kind of cut and slice food. So think like if you're eating an apple, and you take that first bite, you want to like slice off a piece so that you can, chew it some more. That first slicing bite is done with the incisors. All right, next we have a pair of canines on the top and on the bottom. And the canines pierce food, and that function is seen much better in carnivorous animals, which we are not. Right? We are omnivores. We eat some meat and some vegetables, other critters that aren't animals. But because we aren't dedicated meat eaters, our canines aren't super pronounced, so they're used to kind of pierce and hold food, but not again to the level that they would be used in a primarily carnivorous animal where the food is trying to get away. So that's why our canines are not as large. All right, and then lastly we have 2 premolars and 3 molars. Count the wisdom teeth. Right, they are, I haven't done that. There you go. The 2 premolars, 3 molars that are used for grinding. And in physiology, we call the process of chewing and grinding food masticating. And that's what you do, right? Think about the apple. You bite the apple, you slice it with your incisors, and then you push it to the back of your mouth where you can masticate it with your molars and grind it up into a mushy ball of saliva-infused food that you can then swallow. So we're going to get there in a second. But since we're talking about a mushy ball of saliva-infused food, that thing actually has a name in biology. We call that a bolus. That is the scientific name for a blob of chewed food. So we'll be talking about the movement of the bolus in a second. Okay, any questions for teeth?
It's the same on the top and the bottom, they're the same names and functions.
Yep. Okay. either side of the body. So the largest type of salivary gland that we have is found right next to that big masseter muscle on your cheek, and these ones are called the parotids. We then have sublingual salivary glands that are found in the floor of the mouth. So as their name suggests, they are sublingual, they're kind of under the tongue. And then lastly, we have the submandibular salivary glands, which are on the medial surface of the mandible. So now they're kind of like on the underside of your jaw, your lower jaw, your mandible. Yeah.
Do you know what could cause a blockage in a salivary gland?
So there are ducts that you can kind of see here in green that run off of the glands. What could block them? Maybe you had an infection somehow that gotten from like your oral cavity into the duct and it swelled the duct closed. I'm not sure what else would block them. Because otherwise what's coming out of the salivary glands is just saliva, and the saliva is mostly water with some enzymes and some antimicrobial agents mixed in. We'll talk more about that when we get into like the digestive physiology. But there shouldn't be anything big coming through the ducts that would cause blockage. I can look it up and see. I don't know off the top of my head. The questions for the salivary glands. Like I say, we're just focusing on the anatomy. Probably on Wednesday we'll start getting into the physiology, like what is saliva and what does it do. We're gonna get there. Is it appropriate? Cool. So that was the oral cavity and all the stuff there. So now we're going to continue on through our digestive tube. We're going to get to the structures of the pharynx that are specifically associated with the digestive tract, and the esophagus and the stomach. So pharynx we've seen already, where we saw it with the respiratory system. And in the digestive system, we're just focusing on two major areas. These are the oropharynx and the laryngopharynx, right? Because the uvula is flapping back and keeping food from getting up here into that nasopharynx. So we shouldn't get food up there. So the way we're following the progress of our bolus, right? So again, the bolus is just the blob of chewed food. Once you chew up a blob of food, your tongue pushes it into the back of your mouth. It goes first into your oropharynx, and then as it progresses on down, what keeps that food from going from your oropharynx into your trachea? Yeah, the glottis, right? So that little garbage can lid flaps down. Right there it is, right there. The glottis. So that will flap down so that the food doesn't then go toward your lungs. And instead the food will be diverted towards the laryngopharynx and into the esophagus, which is not shown on this picture. So pharynx just plays kind of a minor role as a passageway between the oral cavity and the esophagus. Again, the most notable thing is that because air and food mix in these locations, we have a couple of adaptations to keep the food from going into spots where only air should be. Okay, that's pharynx. So next, we have the esophagus. So the esophagus now is this long muscular tube that extends from the bottom of the laryngopharynx all the way to the entrance of the stomach. So I've already doodled on this a little bit, but you can kind of see that it does go down through the mediastinum, and then it goes through the diaphragm muscle, where it then attaches to the stomach. So at the top we have what's called the superior esophageal sphincter. And right, a sphincter is just a ring of muscle that's wrapping around, usually some sort of tubular structure. So this is our esophagus, right? This would be kind of how the sphincter muscle would be laid out. So it would wrap around, and when it was contracted, it would close off that area. And this muscle is made up of skeletal muscle. And the very, very top part of the esophagus is also made up of skeletal muscle. So this tells us what about the potential for control for this muscle. It can be voluntary, right? And of course we can get involuntary control of it as well. But this way when you swallow, when you kind of push your tongue or push the food to the back of your mouth with your tongue, and you're in that initial phase of swallowing, right? Swallowing, although you can do it without thinking, you can also do voluntarily. And that also kind of helps to, as you voluntarily swallow, kind of prep this first area of the esophagus to relax and allow the food to enter in.
Is that why we have a hard time swallowing vitamins?
Probably.
Because we can control our esophagus.
Yeah. Yeah. And also, I mean, just like, so the bolus is, I should have added soft. Usually when you swallow something, it's already like really soft. So swallowing like a vitamin, especially if it's like a big one, it's hard. And your body is just kind of naturally, I think, opposed to swallowing a hard thing because it knows like, oh, this is a choking hazard. Or this might not be easily digested because it's one solid thing. So it could be also just like psychological in terms of like why it's hard to swallow it. But you can definitely feel it, like if you swallow it and it gets kind of like sort of stuck, you can feel it in that first part of your esophagus, so that when you swallow a few more times and it goes down. That additional swallow might be like that skeletal muscle. Right, so that's the one at the top, kind of linked to the swallowing process, skeletal muscle. So can be controlled involuntarily, but has some potential for voluntary control too. The inferior esophageal sphincter is now the muscle that wraps around the bottom part of the esophagus, right where it meets the stomach. So I'm trying to kind of emphasize that on the picture here. And this one is made of smooth muscle. So this one we have no voluntary control over at all. And that makes sense because you don't really, once you swallow something, you kind of just like forget it. It's like, okay, it got out of my mouth, it's going down, I'm done. But that piece of food has to get all the way down through your chest, where the esophagus is, and then it has to get into your stomach. So all that kind of happens automatically. And the main purpose of that inferior esophageal sphincter is to keep the stuff that's in your stomach from easily getting up into your esophagus. And again, we'll talk about this more in a second. But what's the thing that's in your stomach you don't want getting out? The acid, the gastric acid. And again, we'll talk more specifically about gastric acid when we talk about physiology of the stomach. But just to kind of like emphasize the importance of these sphincter muscles, gastric acid is basically hydrochloric acid, the stuff we use in lab. And hydrochloric acid has a pH of around 1. So it's a really strong, really low pH acid, and that's why the body's being so careful about restricting where that acid can go. Okay, that's all I want to say about the esophagus, other than to emphasize that the esophagus isn't just these sphincter muscles, right? Although they kind of border the top and the bottom of this tube. The esophagus is lined, or I shouldn't say lined, because that means like where the lid is. The esophagus is surrounded with muscle all down its length. So it starts out as skeletal and then it quickly transitions to smooth muscle down the rest of its length. And why might you want this tube to be made up of muscle? What happens if the bowls of food gets to about here and it's like, okay, I'm not going any further, like this?
You start to cough.
No.
Well, you're choking, so and then you want it to like go back up.
So funny story.
Or push
My-
it down.
My dad one time got like one of those peppermint, like those hard peppermint candies stuck in his esophagus. And he tried coughing and stuff, but that doesn't do anything because it's not in your trachea. It's in your esophagus. So moving air doesn't do anything, although like you kind of want to because it doesn't feel great to have something stuck in your esophagus. But it didn't stay there forever and you didn't choke, thank goodness. But what keeps the hard peppermint candy from just like staying there forever? Assuming it's not gonna dissolve. Yeah. Yeah, so because you have mostly smooth muscle surrounding the whole structure of the esophagus, the smooth muscle will rhythmically contract, and those rhythmic contractions, which we'll talk about in a few moments. Where are the next slide actually? Hold on, let me see. Where do we see what my next slide is? Oh, there we go. I do have it on here. So the esophagus, let me just backtrack a smidgen, right, is expandable, so you can get boluses of food of various sizes to go down it. It is surrounded by muscle, which is what I was talking about. And the way that we keep things from getting stuck in there is that the smooth muscle will produce these rhythmic contractions that are called peristaltic contractions, or peristalsis, that are used for the function of movement. So peristalsis, smooth muscle contractions in the gastrointestinal tract are used to move things. Okay, so that was all I wanted to say for the esophagus. And before we get to the stomach, I'll talk a little bit more about peristalsis and then the other kind of major smooth muscle contraction activity that happens. Is everybody okay with this? Okay, so the way that peristalsis works, so there's two major types of contractions that occur in the smooth muscle around our digestive tract. Peristalsis is one of them. So peristalsis is used to move the bolus of food in one direction. So it keeps the blob going, in this case for your esophagus, keeps it going down towards your stomach and not back up towards your mouth. And the way this works is that these contraction waves in the smooth muscle occur behind the bolus of food, and that propels the food blob forward a little bit. And then after it's moved forward a bit, the next contraction area occurs again in a different part of the tube, now once again behind where the bolus is, and that pushes it forward. So it's like when you're trying to squeeze toothpaste out of a tube of toothpaste, right? You start out with the toothpaste tube and it's full, so you squeeze towards the bottom, but then as it gets more empty, you squeeze closer to the top. That's what this is doing. It's just squeezing in different spots depending on where the blob of food is. It's always squeezing behind the blob or the bolus of food. So that's peristalsis. That's how it keeps the bolus always just moving in one direction. The other type of muscle contraction that we see happening in the digestive tract is called segmentation. And in segmentation, contractions happen all at the same time in different segments of the digestive tract. So here you can see the contractions were just in one spot. Segmentation, now they're all happening in different spots but at the same time. So what segmentation does is it doesn't move the food in one direction. Instead, it mixes the food. And during this mixing process, there's no net movement of the food. So the food kind of all just stays in one spot. But you can see, what started out as, maybe first you ate some rice, and then you ate some corn, and then some salad, and then some, what are those, beans, right? Instead of having them all kind of separate from each other, segmentation mixes them all together. And this just helps make sure that everything that you ate gets a chance to contact the mucosa that lines your digestive tract. And we see this mostly in the intestines. So when you're having a lot of chemical digestion, you want everything to be able to touch that intestinal mucosa. you need to kind of mix everything up really good to ensure that that happens. Yeah,
And this is smooth muscle?
and this is all smooth muscle. Yeah. Yeah, skeletal muscle is super rare. So we have it, the very first part of the esophagus is kind of neat. But predominantly we see smooth muscle. Yeah. Other questions? Okay, so next, the stomach. So again, just to kind of orient ourselves, so here's the esophagus right here. And then I already have a little arrow drawn. So this is where our bolus of food will be coming down into. And then it would enter into this pouch-shaped organ, which is the stomach. So the stomach is a very muscular organ, right? Just like the esophagus, which is mostly surrounded by smooth muscle. The stomach is also surrounded by several different layers of smooth muscle. You don't need to know all the names of them, I just pointed them out on here, but you can see they run in different directions. And the fact that we have these different orientations of the smooth muscle in the stomach does mean the stomach plays a role in helping to mechanically digest the food that enters in. So the stomach can kind of contract and press on the food and help to break it up smaller. All right, before we get to the regions, the other physical characteristic we can see of the stomach, besides this really thick outer layer of smooth muscle, is that just like we saw in the bladder, we have these folds that are called rugae. And the rugae of the stomach work the exact same way as the rugae in the bladder, right? Think of these pouch-like structures as being like empty plastic bags. So when you have an empty plastic bag and you want to, carry it around, you fold the bag all up, and the little folds of that plastic bag are really evident. So when the stomach is empty, these folds or these rugae are really evident and present. So if you eat a meal and the stomach fills up, just like as you're filling a bag with materials, the rugae disappear as the stomach expands. And it works exactly the same way for the bladder. Okay, so next we'll get to the regions of the stomach. And we'll start with a region that kind of pokes just up over this gastroesophageal junction, so where the esophagus meets the stomach. We have a little area of the stomach that kind of protrudes superiorly just above that junction, and that area is called the fundus. So the fundus is just superior to this gastroesophageal junction against this area that kind of pokes up like that. All right, so next we have the area also near the gastroesophageal junction. It's more on the medial part of the stomach, and this area is called the cardia. So the cardia is again that superior part of the stomach, it's near kind of the top, and it's within 3 centimeters of the gastroesophageal junction, but it doesn't stick up above it. So that's what makes the fundus different from the cardia. Part above the gastroesophageal junction, the part of the stomach that's right around it is the cardia. And then the rest of the stomach is, before we get kind of towards the end where the intestine is, the major segment of the stomach is called the body. So basically this whole area, this large region of the stomach between the fundus and the pylorus is the body. And then lastly, the pylorus is kind of this triangular or funnel-shaped area of the stomach that's attached to the first part of the small intestine, which is the duodenum. And just like we had a esophageal sphincter, right, keeping food from getting out of the stomach and into the esophagus, we also have another sphincter muscle, which is this pyloric sphincter muscle, that regulates the passage of food out of the stomach and into the small intestine. Because again, stuff coming out of the stomach is super acidic. You don't want to just dump a ton of acidic materials into the intestine. You want to regulate the movement of that substance. And so there's a smooth muscle sphincter there as well to just control the passage of food out. There it is. Okay. So again, this is all just structural anatomy. I haven't forgotten about all the digestion, cool stuff that happens here. But are there any questions for this part? Okay, awesome. So next, get into, I think, my favorite part of the digestive tract, which is the small intestine, because there's just so much cool stuff happening there. And the small intestine, right, is the tube, the GI tract tubular structure. But attached to it are a bunch of accessory organs that aren't part of the tube. So we'll talk about the tubular structure of the intestines, and then we'll also go over the accessory organs that are secreting into the small intestine. So the small intestine is made up of three major areas: the duodenum, the jejunum, and the ileum. So, I need to get my numbers right. So just to give you an idea of the length of these structures, the duodenum, this part that's kind of highlighted in blue here, right? The duodenum, which is the first part that comes off, first part of the small intestine that's attached to the stomach, right? It's not very curvy. The duodenum is around 10 inches long, right? So like you saw in the mink, right? It's not a super long part of the small intestine. And this is receiving the food that comes out of the stomach. Oh, and one last thing. Once the bolus of food goes into the stomach and mixes with all the acid and other digestive juices in the stomach, we don't call it a bolus anymore after that. We call it chyme. So the bolus becomes chyme after it goes into the stomach. So now this is a blob of digested food. And where it starts to be digested is in the stomach. So the chyme leaves the stomach, goes into the duodenum, and the duodenum is attached to our accessory organs. It receives digestive enzymes from the pancreas, which we'll talk about in a little bit, and it receives bile that comes from where? Ooh, you're right, the liver. So the liver stores it in a little pouch first. The gallbladder, right, yep. But good, yep, so it is, so the bile is being made in the liver, it's being stored in the gallbladder as a little pouch, and then it's secreted into the duodenum. So the duodenum gets stuff from the pancreas and the gallbladder also. And again, about 10 inches long. Which, to give you an idea, I'm super jazzed that we've got this giant measuring stick. It's like 5 feet long, so happy, right? So 10 inches is like from here to there. That's the duodenum. Right? Not super long. The jejunum is, by comparison, 8 feet long. Oh, I wrote it extra big. Extra excited about it. So 8 feet of jejunum, which is the second part of the small intestine, where we get a lot of, it's just all coiled up. I know, and that's why the small intestines are getting, in the mink, I was like, don't worry about all this coiled part, like the part that's attached to the duodenum, the jejunum, or whatever. And the reason why it's so coiled is that you can fit 8 feet of jejunum into your abdominal cavity. Okay, so while we're on this, before I start measuring out more feet, the ileum, does anyone want to guess? Oh, not that long. But altogether it would be 20. So it's 12 feet. I guess 12 feet plus 8. 12 feet of ilium. So okay, just to give you an idea, right? That was the duodenum. This is 5 feet to there. So then 5, 6, 7, 8 would be 36. So there is your jejunum. If you stretch it all out. And then if you took that and you added 8, 9, 10, 11, 12, 4 more feet to that, I wrote on a board, but that would then be the length of your ilium. It would be like from here. Let me see, 4 feet? What's 4 feet? 48
inches.
Yeah. I'm gonna be from there. Right there. Oh my goodness. So that's your ilium. And that's all coiled up, making up your small intestine. So both the jejunum and the ilium are actively involved in chemically digesting the stuff that came out of the duodenum. So you're getting continuous digestion occurring from the pancreatic secretions and the lipid digestion that's being assisted by the bile. And then you also have enzymes all along the mucosa of these areas of the intestine. So the major functions here are continued chemical digestion and absorption of nutrients. For the jejunum and the ilium. So why are these structures so long? Right? Like to increase surface area, right? You want the most chance possible for your little bits of glucose to hit the mucosa and get pulled into your bloodstream by those intestinal cells. So the incredible length of these organs is to maximize surface area. Exactly. But we don't stop there because the intestines are all about surface area. So on top of that, the inside of the intestines have these circular folds that are called plica. So that's this over here. So inside.
Is that in all of them?
Yeah, and all of them. Yep. So just pretend this is like, a little tube here, our ilium structure. So there are these circular kind of like ridge-like folds all through. There you go. Like we're looking to like a chunk of it here. These circular ridge-like folds all through the intestine. And then on the circular folds are little finger-like folds that are the villi. So little finger-like projections or folds that stick off of the plica. Like that. And then on the villi are little simple cuboidal cells. So if you took a microscope and you look at the simple cuboidal cells under the microscope, you'd see on their surface that they are folded. Right? And those are the microvilli. So again, like surface area is just done as much as it can be done in the small intestine. We do it by making the small intestine really absurdly long, and then we put little circular folds in it, and then we put little finger-like projections, and you can see it's like little brushes in the car wash. They're just like covering the inside, like little brush-like finger projections. So it's not just a few of them; it totally covers this area. And then there are microvilli on the surface of the cells. So really, really good at absorbing materials that have been digested. Questions about that? Okay, cool. we get the accessory organs. So these are the organs that are not part of the tube, but that are secreting into the tube. So again, just to orient ourselves, here's the tube right here. Here's our duodenum. I outline it right there. Okay, so the duodenum sits kind of underneath the liver. And then right in that first curve of the small intestine, which is here, we have this bumpy yellow organ, which is the pancreas. Now I'll just circle. So the liver secretes bile into the duodenum by way of the gallbladder. So there's a little duct that comes off the gallbladder and allows its secretions to enter into the small intestine. And then that duct joins up with a pancreatic duct that also allows pancreatic secretions to enter into the duodenum. They both kind of just get released in at the same spot. Okay. So that's just to kind of give you an idea of like where everything is. So what I want to do next is talk a little bit first about the liver, and then we'll talk a little bit about the pancreas. Let's do the liver first, because why not? It's more superior, it's really big, I don't know, there are a lot of reasons. Let's talk about it first. So the liver, right, you see in lab, we saw it in the human models, so you're kind of familiar hopefully already, that it occupies most of the right hypochondriac region and into a bit of the portion of the epigastric region as well. So it's really kind of occupying the right side of the body, just underneath the ribs, and then a little bit towards like your midline. One part of the liver structure that we've seen already when we did blood vessels is the hepatic portal vein, which is really, really important because it takes blood from the mesentery of the intestines and from the capillaries around the stomach and carries that blood into the capillary beds of the liver before putting that blood back into like general circulation towards your heart. And this is important due to a couple of things that the liver does. So there's a few reasons why we want the liver to have first dibs on this fairly nutrient-rich blood that's coming out of the intestines and the stomach. One is that the liver provides some protection. So cells in the liver have really, really big, smooth endoplasmic reticulums, and that allows the liver cells to be really well adapted at detoxifying poisons, like alcohol, right? So people who overconsume alcohol are at really high risk to damaging their liver. And that's one of the reasons why, because the blood that comes out of your intestines and your stomach goes to your liver, and then your liver is trying to take that alcohol that's been consumed or any other toxins and convert it via metabolic processes into something not toxic or less toxic. The other thing the liver does is it modifies nutrients before they go into the vena cava. And in some cases, it can take things like glucose and convert, say, the glucose into glycogen, which is like our body's starch, right? We're not potatoes, so we don't make starch. But we make glycogen. We store excess carbs that we don't turn into adipose tissue. We can turn them into glycogen instead and store them in our liver cells. So that's just an example of like how we might modify a nutrient and store it more before it goes into the vena cava and into general circulation. Okay, so continuing on, right, the other thing we know the liver does, it's important to the digestive system, is that it produces bile from the breakdown products of destroying the hemoglobin that comes out of our worn out red blood cells, right? So the liver makes bile, and then that bile that's formed is stored in this green pouch that's called the gallbladder. And the gallbladder holds that bile until hormone signals tell it to release that bile. So there's a little hepatopancreatic sphincter that stays closed and prevents bile from being secreted into the duodenum until there are appropriate chemical and usually hormonal signals that cause the sphincter muscle to relax, and then that allows the bile secretion to go into the duodenum. So bile is really just used for the digestion of one specific group of nutrients. It's really just used for the digestion of fats. And again, we'll talk much more about this when we do digestive physiology next time. But what it does is it emulsifies fats. So if you have like one big blob of like oil or fat, it's really hard to digest the triglycerides in the middle of that blob. If you're a little enzyme that's working on the outside of that blob, right? Getting to here can be really challenging. So what the bile does is it takes that big blob of fat and it breaks it into lots of little blobs of fat. And now once you have a bunch of little blobs, it's way easier for those enzymes to get all the way through and digest all the triglycerides in this blob. So that's what it means in increasing surface area. It's taking big fat blobs and turning them into lots of little ones. Okay, and I think that's all I want to say for now about the gallbladder and bile. Okay with that? Yeah.
What's the view of that liver? Is that inferior?
That's inferior, yep, so it's looking at it from the bottom. Okay, so our other accessory organ to the small intestine is the pancreas. And we've gone over its location many, many times. Just a reminder, right, it's in that first curve of the small intestine, right under the stomach. And pancreas we've seen before because we talked about the pancreatic islets. And the pancreatic islets have nothing to do with digestion, right? Pancreatic islets are involved in making hormones. So the pancreas kind of has two major jobs. One job is to make hormones that are primarily concerned with controlling blood sugar levels. The second job of the pancreas is to have this exocrine function, where it's not putting its secretions into the bloodstream. In this function, it's putting its secretions right into a little duct. And then that duct, in this case, is emptying right into the duodenum. And then the secretions are acting there and then throughout the intestines. So in this exocrine function, the cells that we're looking at are called pancreatic acini, and they're also called, I'll on the board, write that down. They're also called acinar cells. I think because they look like little berries. But these are just clusters of secreting cells that make digestive enzymes. And when I say they make digestive enzymes, I mean to digest every kind of organic molecule you can imagine. We have proteases to break down proteins, nucleases to break down nucleic acids, amylases for big carbohydrates, and lipases for lipids. So pancreas is really playing an important role in digesting the various nutrients that you consume. And again, I highlight the type of cells just to drive home. This is a different group of cells than the pancreatic islets that we've talked about in the past.
What did the nuclease digest?
Nuclease, nucleic acids.
Okay.
Yeah, so if there are any nucleic acids in the food that you've eaten, you can actually break those down. Kind of cool. Right, and this is just recap of the my things up here. So other pancreatic juice information, right? Secretions go into ducts. Again, secretions don't go into the bloodstream the way the endocrine cells of the pancreas do. And the other duct cells make the other secretion materials. So besides enzymes, we also get some water and some bicarbonate. So bicarb we've talked about in the plasma, the blood plasma in the past. We also get bicarbonate doing a similar function to what it does in the blood plasma, which is buffering. And that increases the pH to around 8. Instead of what's coming out of the stomach, right when the chyme first comes out, which is a pH of around 1. So the reason that happens is because all those nice enzymes on my previous slide, they don't function at a pH of 1. So the pancreas has to secrete a buffer to raise the pH to around 8 or so, at least around like neutral, like around 7 or 8, to get those enzymes to work. And then those go into the duodenum when you relax the same sphincter muscle that allowed the bile to get in. So again, there's a common tube that both ducts join up with, the duct from the gallbladder and the duct from the pancreas. And then when you relax that hepatopancreatic sphincter, that lets both the pancreatic juices and the bile into the duodenum. Okay, so last thing I'll talk about is, for this anyway, what controls that hepatopancreatic sphincter? Like, how does the body know whether it should be saving its bile because no food is passing through that area just yet, or whether to relax that sphincter muscle and release the bile and pancreatic secretions? So under normal resting conditions, if you haven't eaten anything recently, that hepatopancreatic sphincter muscle stays closed until something stimulates it to relax and allow the bile and pancreatic secretions to go into the duodenum. So like I mentioned, this is primarily due to hormonal controls. These are chemical messengers that get into the blood. And this mostly happens like when you've eaten something and the presence of nutrients stimulates the release of these hormones. So one of these hormones, CCK, or, yeah, I don't want to mess up my pronunciation, cholecystokinin. Cholecystokinin comes from your intestinal cells when they get exposed to fats and proteins in the chyme. So chyme comes out of the stomach, goes into the duodenum, the cells of the duodenum get exposed to these fats and proteins. There are receptors on the cells that bind to those nutrients, and then certain cells, as a result, will secrete this hormone, this cholecystokinin, into the bloodstream. And this stimulates the gallbladder to release bile. And it also stimulates relaxation of that hepatopancreatic sphincter muscle so that both the bile and the pancreatic secretions can get into the duodenum. Yeah.
You said it stimulates being relaxed?
The sphincter muscle, yeah. So when the sphincter muscle is like in its resting state, like nothing is stimulating it, it's contracted. And it's like pinching closed the bottom of the duct that lets the bile and the pancreatic juices into the intestine.
Is that the feeling of hunger?
No, but if you're hungry, that muscle is closed because there's nothing actively being digested. Like unless you've recently eaten and there's food moving into your intestine, there's not going to be anything to stimulate that muscle to relax. So literally just like the presence of nutrients in your small intestine, that's really what's like kicking off the stimulation to relax that muscle. So if you haven't eaten anything for several hours and your beginning part of your intestine is not containing food, that muscle is contracted. Okay? Very cool. Other hormone I'd like you to know is called secretin. So this is going to be, again, released by intestinal cells into the bloodstream when intestinal cells get exposed to gastric acid. Because gastric acid presence tells your body that, chyme, and not just acid, but all the nutrients that are also in it are moving into the intestine, and now is an appropriate time to cause the gallbladder to release bile, and cause secretions to be released into the intestine. Okay. Other chemical controls include circulation of the blood, returns most of the bile salts from the ileum back to the liver. So we don't just release most of our bile salts that we use to digest things out with the fecal material. Most of those bile salts come out of the ileum and go back into the bloodstream, and they're carried back to the liver. And when we have a large amount of bile salts going back to the liver because you've been digesting a lot of food, that also stimulates the gallbladder to release bile. That's a way of your body kind of understanding that, a lot of food's coming through here, probably time to release more bile. And then lastly, we have neural control, right? So parts of our parasympathetic nervous system are attached to the gallbladder. So specifically the vagus nerve, right, which is part of the parasympathetic nervous system. It goes from your medulla all the way down into your abdominal cavity. Axons from the vagus nerve also stimulate the gallbladder to release bile. So your parasympathetic division is active when you're in your rest and digest phase. And so this is a lot of text, right? This is right from figure 23.29. This is all the same thing I just talked about, but in a picture. So pictures are better for your brain. Pictures are better for my brain anyway. This kind of helps to summarize the different processes that are helping to control bile secretion and also relaxation of the hepatopancreatic sphincter to let materials into the duodenum. And it also ties in the bloodstream, so you can even see, like the movement of the hormones, at least for the cholecystokinin and the secretin. I think it also shows the nerves as well. Yeah, and the bile recovery. So yeah, so if pictures are helpful for you, check out this figure. It's right from Silverthorne. And that just summarizes all the things I just talked about. Questions about any of that? Okay, so then last thing we'll look at are just the large intestine and the mesenteries, because there's not a ton going on there. So we use that to wrap up our exam-free material. So the large intestine, when you look at it, right, the large intestine is way less coiled and twisty than our small intestine. And you see kind of all along the large intestine that there are these little pouches. They're called haustra, right? We saw those in lab as well. So the haustra are, again, pouches that basically just store digestive material, and they also allow for expansion of the large intestine. So these are pouches that are increasing surface area for absorption, the way we have all these little C-shaped projections in the small intestine. These are just there for storage and to accommodate the mostly digested food material that's entering into this space. The other thing we see on the large intestine is this longitudinal band of muscle that kind of runs right through the center of all these haustra. And this is called the taenia coli, right there. And this is just, again, they have a smooth muscle that contracts to propel food through the large intestine by peristalsis. So then for our large intestine, in terms of like actual structures, right at the beginning of the large intestine, we see this kind of square-shaped dead-end structure that's called the cecum. And the cecum is the area where the ileum contacts the large intestine. So here we find this ileocecal valve that controls movement of food materials out of the small intestine and into the large. In humans, coming off of the cecum is a little tail-like structure, like that, and that is the appendix. So the appendix is often referred to as a vestigial structure that doesn't have any function other than to get infected and cause appendicitis. Now further research, especially into the microbiome, suggests that that's not true. It is looking more like the appendix is acting as an area that produces a helpful bacterial reserve for your large intestine. So we'll talk about it more when we do physiology, but your large intestine has a lot of bacteria, especially E. coli in it. That's good E. coli. It's the E. coli that helps us digest things that we otherwise couldn't digest ourselves. And the idea is that the appendix kind of stores extra of those bacteria. So if you have like a really bad stomach bug and stuff's moving through your large intestine really fast, you don't lose an enormous number of those helpful bacteria.
So if you do have appendicitis and you do get your appendix removed, you get hit with that stuff a lot harder?
I don't know that they've shown that it makes a huge difference, but like presumptively, yeah, that could happen. That you wouldn't have as good an ability to replenish your microbiome when you've got a really bad stomach bug. It might take longer. Okay, literally the last thing, got 1 minute. Last thing is just the major structures of the large intestine, and then we'll call off today. And again, this is mostly review. So the large intestine, right, starts with the cecum, the little dead end structure where your small intestine is attached. Then the food material goes up the ascending colon. And we didn't see this in the mink, but in humans, the ascending colon is attached to a transverse colon that goes horizontally across the body, and then down into a descending colon, which we also saw in the mink. The last part of the colon is called the sigmoid flexure, or the sigmoid colon, which is basically just where the large intestine makes kind of a little S-curve, like that. And then food goes into the rectum, where it is stored until it is excreted out of the anus. And the anus, like the urinary bladder, has two sphincter muscles. The internal, that's made of smooth muscle that you can't control, and then external, it's made of skeletal muscle that you can. The end. The digestive system. So that'll be the last new material for exam 3. So I'll update our learning objectives and the videos, things like that. And I should have the assignment for you to start looking up. The original email I sent. You know what, it probably went into my junk folder, because it didn't have going back that